Hello dear students, this is Praful and I welcome you all to One Page Biology. In today's video, I will be covering one important question which had appeared in CBSE 2018 biology paper. So let's read the question. The question is, write the scientific name of the organism Thomas Hunt Morgan and his colleagues worked with for their experiments. Explain the correlation between linkage and recombination with respect to genes as studied by them. Now dear students, as you can see, this question had appeared in 2018 paper and this question was precisely asked for 3 marks. There was another question which was 2 marks, which is based on the scientist Sturtevant and his findings which were related to gene order and gene frequency. So we will try and make another video on gene order and gene frequency. In this video, we are going to mainly focus on what exactly is linkage and recombination. As we all know that Thomas Hunt Morgan came out with certain findings related to the linkage as well as recombination. Now he was born in the year 1866 and he died in the year 1945. Dear students, he was also known as an American revolutionary biologist because he made certain observations which were related to linkage and recombination, and these observations were very much new during that time. Also dear students, he is a genetist, he is embryologist, as well as he wrote many books in the field of biology, and that's why he is also known as a science author. So dear students, T.H.Morgan received Nobel Prize in the year 1933 for discovering the role of chromosomes in heredity. Now as far as the experiment is concerned, dear students, T.H.Morgan had carried out the experiment on fruit fly. The scientific name of fruit fly is nothing but Drosophila melanogaster. Now dear students, T.H.Morgan carried out an experiment in which he allowed the two Drosophilas to mate with each other. Now in this particular case, he used a male Drosophila and a female Drosophila. Now the male Drosophila was brown body red eyed and the female Drosophila was yellow body white eyed. So he allowed them to undergo mating, and after the mating, he got the F1 generation. So F1 generation stands for first filial generation. That means that is the first generation which is formed after the parents undergo mating. Now dear students, Morgan observed that all the F1 generation Drosophilas had the characters which were exactly same as that of the parents. So now he allowed the F1 generation to mate with each other. So he got the F2 generation. Now dear students, T.H.Morgan observed that 98.7% of the Drosophilas were exactly same as that of the parents. So these were considered as the parental types, whereas 1.3% of the Drosophilas were slightly different from the parents, so they had mixed characters. So these Drosophilas were considered the recombinant types. So from this particular results, he said that because maximum type of the F2 generations were getting the same characters which were there in the parents, from that he said that the characters of the body color as well as the eye color are showing linkage. Now dear students, that means that the genes which are coding for these particular characters are mainly situated close to each other, and as a result of which these genes are continuously getting transferred to the next generations. So as a result, these genes are showing very high type of linkage. In the second part of the experiment, he had considered the second characters. Now in this particular experiment, he had used red-eyed normal wing male and white-eyed miniature wing female. Dear students, here the word miniature means small. Now again, in this particular experiment, he observed that all the F1 generation Drosophilas were same as that of the parents. Now, as far as the F2 observations were concerned, he observed that in this particular case, 62.8% of the Drosophilas were exactly same as that of the parents. That means they were parental types. But dear students, in this particular case, 37.3% of the Drosophilas were different than the parents. That means these were the recombinant types. So dear students, that means the percentage of recombinant types was very high in this case. So from this particular observation, Morgan interpreted that the characters of wing shape and the eye color are located far away from each other, and because these characters are located far away from each other, they are showing less linkage and high recombination. Dear students, another very important scientist, that is Alfred Sturtevant, he was a student of T.H.Morgan. Now he came out with certain laws regarding gene recombination. Now dear students, he said that the frequency of gene recombination is directly proportional to the distance between the genes. That is, higher the distance between the genes, higher will be the recombination frequency. So dear students, That was about linkage and recombination in this particular video. I hope you understood the concept of linkage and recombination. If you have any doubts or suggestions, please put it in the comment section. You can also mail me your suggestions on my email ID, which I have put it in the description box. So dear students, if you have understood the concept, please like, share and subscribe to One Page Biology. This is Praful signing off. I'll be seeing you all in the next video with some another biology related question. Till that time, take care. Thank you so much. Bye bye.